Uh, Caitlin Baker, please, would you like to share your screen?
All right. Let's see.
And
is everything visible?
Yes, very good. And Caitlin will talk about population genomics of Charinus whip spiders from Israel. Very good.
Yes.
Go on.
Thank you. Today I will be telling you about some work that I'm doing in my postdoc, looking at the phylogenetics and population genomics of a couple of species of Charinus whip spiders or amblypygids in Israel. And this is part of a much larger project that's being done in collaboration with members of Efrat Gevish-Regev's lab and Prashant Sharma's lab. And we're only in the first year of our multi-year grant studying the speciation dynamics of Israeli cave arachnids and the genetic basis of arachnid development. So do stay tuned for more exciting results related to this project coming in the near future. So the focus of our grant is on Israeli cave arachnids, and Israel is situated in the Levant region. This is an interesting biogeographical theater because it sits at the juncture not only of 3 different continents, Europe, Asia, and Africa, but of 3 climatic zones as well. There's Mediterranean, steppe, and desert climates. Furthermore, during the Pleistocene, the glaciers that covered much of Europe didn't reach as far south as the Levant, which allowed its mesic climate to serve as a refugium for different taxa. And this is thought to have contributed to the high endemism in the region. And in particular, it is the caves in the Levant that are suspected to have acted as refugia given their stable temperature and humidity. Now, caves are a textbook example of an arena of adaptive evolution. Across many different groups of animals, we see the same phenotypes repeatedly evolve in cave-dwelling species. The lack of light is a really strong selective force that positively selects for features that give advantage in darkness, such as appendage elongation or increased olfactory capacity. But the reduced reliance on vision can also lead to things like the atrophying of eyes, where they become smaller or in some cases disappear entirely. And much of what we know about the process of cave adaptation in animals comes from 2 model systems, the teleost fish Astyonyx mexicanus and the isopod Acellus aquaticus. There have been multiple independent colonizations into cave habitats in both of these species, and developmental biologists have leveraged this parallel evolution to interrogate whether the same specific genetic pathways produce a particular cave phenotype across different populations. But this is the arachnology meeting. I'm here to talk about arachnids. What do we know about the process of cave adaptation in arachnids? And the short answer is not much. But this is where our focus on Israel really comes to the fore, because here we have a rather unique system for probing just these kinds of questions. We have 3 instances of arachnids in Israel, wherein we have a pair of sister species that exhibit contrasting morphologies. One is eye-bearing or epigean, and the other has reduced eyes or is hypogean. So rather than focusing on a single taxon like the fish or isopod species, this system allows for a comparative approach. We have multiple data points from a single geographic region, and we will be able to determine if they show similar biogeographic patterns. And if they do, this could indicate a shared or common mechanism driving the evolution of Israeli arachnids into cave environments. So knowing the phylogenetic and biogeographic patterns will also inform the questions that we ask about convergence or parallelism in the development of eyes in these groups. So for this talk, I'm only going to be focusing on the amblypygids, but we are hard at work generating similar data sets for the other arachnid groups as well. In Israel, there are 2 species of amblypygids. They're both in the genus Charinus, and there's Charinus ioneticus and Charinus israelensis. Charinus israelensis was relatively recently described in 2016 by Miranda et al. On the basis of morphology. And it is readily differentiated from ioneticus by its highly reduced median eyes. The 2 morphospecies also have very different geographic distributions. Ioneticus has a broad synanthropic range across the Eastern Mediterranean, whereas Israelensis is only known from 2 localities in northern Israel. And here is a map of their distributions just to illustrate how widespread Uneticus is across this region. Within Israel, Uneticus is typically found in humid, man-made habitats like cisterns or the walls of old buildings. And the 2 known Israelensis localities are also humid, man-made caves. One final point of interest, for both of these species, only females have ever been found. And this has led to the suggestion that both of these species are parthenogenetic or reproducing clonally. So with all of that as background, I'm going to address 3 questions in my talk today. First off, is Charinus israelensis a valid species? That is, does genetic data correspond to morphology? I'm also curious how geographically structured populations of Charinus are in Israel. Amblypygids are often heralded as examples of short-range endemics, so does that pattern hold here? And then finally, how many times has troglomorphism or cave-adapted morphology evolved in the Israeli Charinus? And to answer these questions, we need to have a robust understanding of exactly how many species there are, their phylogenetic relationships, and we want to know something about their genetic diversity. So to address this, I extracted DNA from 40 Charinus individuals from 5 localities in Israel. These specimens were all collected in 2018 by my co-authors from the Sharma and Gavish Regev labs. They did look in many other caves all over Israel and Palestine while collecting other arachnids, but these are the only places where they found Charinus. And we prepared genotype by sequencing libraries, GBS libraries for all of these samples. And after getting the sequence data back, I assembled them de novo using the program IPyRAD. I'm gonna skip through most of the methods here, but rest assured, we did have very high sequencing coverage across all samples. The average number of loci recovered for each individual in my final matrix was over 30,000, so it's a very rich data set. So using the loci generated in IPyRAD as well as the SNP data by itself, I performed phylogenetic analyses in IQtree, and I recovered 2 extremely well-defined clades. And these largely corresponded to the morphospecies identifications, which is great. I recovered the same tree regardless of whether I used SNP data or the loci as my input, or if I allowed more or less missing data. So that indicates that these clades are extremely robust. And within each clade, the branch lengths were close to 0, suggesting very little genetic diversity within a species. One exception to this is an animal from Mimlach, which is labeled number 35 down at the bottom here. It's quite distinct from the other specimens of Charinus israelensis. And this locality, Mimlach, is also the one place where the species were found to co-occur. Now, I also performed some genetic diversity analyses on a data set that I constructed by pulling 1 SNP from each locus. And one of these analyses was structure. I found that the optimal number of clusters was 2 in this data set, and they corresponded to the 2 phylogenetic groups that I recovered. I also found almost no admixture between those 2 species. Furthermore, I ran an AMOVA, or an analysis of molecular variance, and I found that over 95% of all of the genetic variation in my data set occurred between the 2 species. I also calculated the levels of heterozygosity in both species, and they were very low, near 0, indicating incredibly low genetic diversity within the species. And the inbreeding coefficient for both species was near 0, suggesting there really isn't any cryptic population structure here. So this really low genetic diversity supports the hypothesis that both of these species are parthenogenetic. Now, going back to the structure results, though, that analysis did suggest some admixture between Uneticus and israelensis in 1 sample. And that is that kind of weird sample of israelensis, number 35, the one in my phylogeny that was genetically distinct. It seems unlikely that this admixture would be a result of introgression, though, because, as I said, these both seem to be parthenogenetic species. So one possibility is that this sample reflects an ancestral polymorphism. And interestingly, when I looked back at the animals to check their morphology, this specimen had eyes. It was the only sample of Israelensis with fully developed eyes. So I'm going to show you the same phylogeny that I put up before, only now I've color-coded the terminals according to eye morphology. And I've got a picture of a carapace of Charinus here. I'm going to show you some zoomed-in photos of the median eyes. As I said, in Israelensis, almost all of the specimens have reduced eyes or have lost them completely. But as I mentioned before, there is that one weird Israelensis specimen, number 35, that is eye-bearing. Meanwhile, in Untitled, almost all of the specimens had fully developed median eyes, but one specimen had reduced eyes. It's not as reduced as those of Israelensis, but it still suggests that eye development is quite plastic in both of these species. So just to wrap up, I did find that Charinus israelensis is a well-supported genetic species. It corresponds well with morphology. And within Israel, both species of Charinus seem to exhibit panmixia. There was almost no population structure despite geographic distance, and there was very low genetic variation within each species, suggesting that they are parthenogenetic. And finally, eye morphology is highly plastic, despite the fact that these individuals within a species seem to be genetic clones of each other. Eye reduction has occurred at least once in each species. There are still some things that we're working on with this system. For one, I'm still running a divergence staging analysis to estimate how long ago these two clades diverged. And that temporal information is going to be key for understanding the biogeographic history of this group. A chronogram is also going to be important if we want to illuminate the mechanism by which Israelensis and Untitled speciated. But the population genetic results point to this interesting puzzle. How does speciation proceed in a parthenogenetic species? Each of these species is clearly capable of dispersal, given that the populations are panmictic, which makes allopatric speciation seem less likely. And while they do have divergent morphologies, which might suggest ecological divergence in sympatry, as I mentioned earlier, both species are found in the exact same kinds of niches. They're both found in humid man-made caves. So this is a very open question. And then finally, it would be interesting to know whether the reduction in the median eyes of Israelensis and that sample of Untitled are occurring via the same developmental pathway. As I mentioned at the beginning of my talk, the other big part of this collaborative project is looking at the developmental genetics of arachnid eyes. And we are currently hard at work figuring out the retinal determination gene network in some model arachnid species, which will lay the groundwork for answering this question. And with that, I would like to thank all of my co-authors in the Sharma and Gavish Regev labs, everyone else in the Sharma lab, the facilities at University of Wisconsin-Madison that helped me with library preparation and sequencing, despite the pandemic raging all around us, the organizations that provided permits, and of course, our funding sources. And with that, I can take any questions.
Very good. Thank you so much. We do have indeed time, and we have a few questions already. The first one is, how can you be sure they are actually sister species? Did you include any other Charinus species in the analysis?
Oh, you're letting me use my backup slide. Yes, so we have CO1 data from some other species of Charinus outside of Israel, and that showed that these two species are reciprocally monophyletic, so we were able to root the phylogeny there.
Then we have a comment. It may be an advantage for some experiments if there was intraspecific variability, isn't it?
What was the question exactly?
I think it was more of a comment that it may be an advantage for some experiments if there was intraspecific variability.
Yeah. It's certainly an interesting question of how you're able to get such different morphologies with such little genetic data, or difference, excuse me.
Then we have another question. The troglodyzation processes favor one direction of polymorphism, maybe?
Typically, what you see is the colonization of cave habitats from surface habitats. One of the things that our collaborator, Gustavo Miranda, is working on is how populations of Uneticus from outside the country of Israel are related to the Israeli Charinus. So it's possible that including that kind of information might help us polarize the direction of troglomorphism more robustly.
Very good.
Yeah.
Thank you. Yes, we have a couple more questions, but I think we have to defer them for later. So we move